Hi everyone, this is Ms. Romani, and during this lesson we will learn about gene therapy, a fascinating and promising area of scientific research and biotechnology. If you recall from our introduction to genetics lesson, genes are made up of DNA. As far as we know, humans have between 20,000 to 25,000 genes. With a few exceptions, we have two copies of these genes, one that came from our mother and one from our father. These genes contain the instructions for building all the proteins in our bodies, but sometimes people have inherited versions of genes that can result in genetic disorders. And until recently, when somebody inherited a genetic disorder, there was nothing that could be done to change the genes that caused it. You could treat the symptoms of the genetic disorder, but the genes themselves could not be changed. This is where genetic therapy or gene therapy comes in. Gene therapy is the still mostly experimental introduction, removal, or change of genetic material into the cells of a patient in order to treat a specific genetic disorder. The introduced or changed genetic material changes how a protein or a group of proteins is produced by the cells. It can reduce the level of disease-causing proteins, it can increase the number of disease-fighting proteins, or it can produce new or modified proteins within the cell. And most gene therapy is still at a research stage, but the process of gene therapy is advancing tremendously and very quickly. So in 2015, there was an international summit on human gene editing in Washington, D.C. And at the end of the summit, its organizing committee released a much anticipated statement recommending how human genetic engineering should be regulated for the future. In their statement, the committee was generally supportive of human gene editing, as long as it is used for somatic genetic modification. In other words, gene therapies that affect only the body cells of the treated individual and not their future offspring. The clinical use of germline modifications, those that would be in every cell of a resulting baby and be passed on to future generations, was not approved. Germline gene therapy is what happens when DNA is transferred into the cells that produce reproductive cells like eggs or sperm in the body. Germline therapy can also involve editing of early embryos. And this type of gene therapy allows for the modification of genes that are heritable, that is, genes that are passed down from generation to generation. Germline gene therapy is currently not legal in Canada and many other countries, because the risks and ethical concerns still appear to outweigh the benefits. So at the moment, the vast majority of gene therapy is performed on somatic cells. So how does gene therapy work? Well, there are several approaches to gene therapy. I will discuss three during this lesson: gene augmentation, gene editing, and gene inhibition. Let's start with gene augmentation. Gene augmentation therapy is often used to treat disorders caused by a mutation that stops a gene from producing a functioning protein. For example, people with a disease called cystic fibrosis are not able to make a functioning protein called the CFTR protein. The CFTR protein is a channel protein that conducts chloride ions across the cell membrane, helping to maintain the balance of salt and water on many surfaces in the body. And it is the lack of a functioning CFTR protein that causes cystic fibrosis. And people with cystic fibrosis lack a functioning CFTR protein because the gene that codes for the protein has a single mutation. So gene augmentation therapy involves inserting a strand of DNA with a normal functioning allele or a normal functioning version of a gene into the cells of an organism. In the case of someone with cystic fibrosis, it would involve introduction of a normal version of the CFTR gene into their body cells. Very often, a carrier molecule called a vector is used to deliver the gene, and the most common type of vectors are deactivated and genetically altered viruses. So in order to use a virus for gene therapy, the first thing that needs to happen is that their viral DNA needs to be removed. This leaves behind only the viral capsid. Without their genetic material, the virus cannot cause disease. However, the viral capsid can still enter our body cells. So once we have an empty viral capsid, then the gene that needs to be inserted into the body cells for gene therapy is inserted into the empty viral capsid. At this point, the virus is called a vector, and these gene-carrying vectors can then insert the gene into cells in two different ways. One is called in vivo gene therapy, and the other ex vivo gene therapy. During in vivo gene therapy, the gene is directly inserted into the body cells, often by injection or sometimes by inhalation of the virus. The inhalation technique, for example, has been used to deliver cystic fibrosis gene therapy into the cells of the respiratory tract. During ex vivo gene therapy, the whole experiment of gene transfer is performed outside of the body. Cells from the patient are grown in a petri dish, then they are genetically modified with the viral vector and multiplied in a lab. And once they have been successfully genetically modified, they are inserted back into the patient. Gene therapy may sound simple on paper, but in reality, there are many challenges associated with this biotechnology. And different vectors, as you can see in this diagram, can create or reduce some of these challenges. One of the challenges involved delivering the genes into the right cells. That is, the cells of the body where the gene therapy will be most useful at treating the genetic disorder. There's variability in how effective the gene is delivered to cells, how many cells need to have the gene therapy for the therapy to work, and how much of the target protein the introduced gene manages to produce. Another challenge is the longevity of the therapy. In other words, does the gene therapy produce a permanent treatment, or does it have to be repeated throughout the patient's lifetime? Since most gene therapy uses a viral vector to introduce genes into the body cells, one other major challenge is the body's own immune system and our body's own immune response to the therapy. The viruses used in gene therapy are naturally occurrent and mostly benign. Because of this, individuals who have already been exposed to the specific viral vectors in the past will have already developed antibodies against those viruses and will be unable to receive gene therapy using that particular vector. And even for patients who have never been exposed to that virus in the past and do not have immunity to it, there will be an immune response to the new introduction of that viral vector. After all, that's what our immune system does. It responds to viruses. And so depending on the viral vector involved, and oftentimes also the patient involved in their own immune system, there could be either a low immune response or a much stronger immune response to the vector itself. And although the last decade has seen major advancements in this type of therapy, gene therapy has been around for a while. For example, in 1990, the first genetic disorder was treated successfully with gene therapy. It was in a 4-year-old girl suffering from adenosine deaminase, or ADA, deficiency, an inherited disorder that damages the immune system and causes severe combined immunodeficiency, or SCID. SCID is a condition that causes bone marrow cells to be unable to produce a vital enzyme, and as a result, people with SCID lack virtually all immune protection from bacteria, viruses, and fungi, which, as you can imagine, can be very serious and life-threatening. So the gene therapy used to treat these conditions was an ex vivo retrovirus therapy that introduced the gene into the patient's T cells outside of the body. Those genetically modified T cells, which are a type of immune cell, were then reintroduced into the patient where they were able to produce the missing enzyme, essentially treating the condition. And in 1993, the first experimental gene therapy treatment for cystic fibrosis was conducted. Like I mentioned earlier, people with cystic fibrosis have a mutation on the gene coding for the CFTR protein. So researchers modified a common cold virus called an adenovirus to act as the vector, one that was carrying the normal genes for the CFTR proteins. The virus was targeted to the cells in the airways of the lungs and was introduced through the nose. This first attempt at treating cystic fibrosis through gene therapy was actually not very successful. There was a low uptake of the gene therapy by the cells, and the treatment was not permanent. However, scientific breakthroughs in the past 10 years have accelerated advances in gene therapy, and better vectors and other innovations means that gene therapy for cystic fibrosis may be available for patients in the near future. So, while augmentation research continues to advance, a new approach to gene therapy is gaining popularity. Rather than introducing the therapeutic gene into cells, a different strategy would be to directly correct the genetic mutations on the existing genes. In other words, editing the gene so it no longer carries the mutation. This approach is gaining popularity thanks to the relatively recent discovery of CRISPR-Cas9. Now, other gene editing tools have emerged in recent years, but none seem to match the precision, low cost, and, well, general usability of CRISPR, which since its introduction in 2012 has been rapidly transforming genetic research and has even entered testing as a medical treatment for some genetic conditions. CRISPR-Cas9 is a simple two-component system used for effectively targeted gene editing. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It is basically a strand of RNA that is combined with a protein called Cas9. The CRISPR-Cas9 method for gene editing is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of viral DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a type of enzyme that can cut DNA at a very specific place that has been programmed by the guide RNA. When the matching sequence on the guide RNA finds a target within the viral genome, the Cas9 protein cuts the viral DNA at that spot. By cutting the viral DNA, the bacteria can disable the virus and be protected from viral infection. So researchers realized that this system could be used to cut not just viral DNA, but any DNA sequence at a precisely chosen location. All that needs to be done is to program the guide RNA to match the target sequence. So once inside the nucleus, the Cas9 proteins attach to the target DNA, find the genetic sequence that matches its guide RNA, and use two tiny molecular scissors to cut the DNA. Once the CRISPR system has made a cut, the gene can either be silenced or edited. To edit a gene, a piece of DNA that carries the desired sequence can be added as a template. The DNA pairs up with the cut ends, replacing the original sequence with a new version. CRISPR-Cas9 is very versatile and can be used in vivo, ex vivo, and even in fertilized eggs, which brings us to a major concern for this biotechnology: the use of CRISPR to genetically modify embryos and its potential use to produce designer babies. A very large part of the scientific community approves genetic therapy in somatic cells, especially in cases of severe disorders like cystic fibrosis. However, the possibility of genetically modifying germlines has been the object of heated discussion. To edit germlines for the sole purpose of enhancing non-disease traits in order to create designer babies is an even more hotly debated topic. One that not only brings up issues of safety, but also issues related to equity and justice. And that is because this type of germline therapy will undoubtedly be available mostly to the wealthy, potentially giving children born to wealthy parents not just a financial and class advantage over poor children, but also a genetic one. And there's also the issue of consent if embryos, who of course cannot consent to gene therapy, are edited prior to their birth. Another area of concern with gene editing are the ongoing experiments to alter the DNA of disease-spreading insects, in part because of the potential unintended impacts the release could have on the environment. Of particular concern is gene drives. Gene drives use CRISPR to insert and spread a genetic modification through a population at a higher than normal rates than natural inheritance. Once a gene drive is engineered into an animal's genome, the animal's offspring will inherit the drive on one chromosome and will inherit then a normal gene on the other chromosome from its other parent. During early development, the CRISPR portion of the drive cuts the other chromosome, and the cuts are then repaired using the drive as a template, leaving the offspring with two copies of the modification. Researchers are currently working on using CRISPR and gene drives to eradicate malaria-carrying mosquitoes and other pests, including invasive species. And finally, let's briefly talk about a third approach to gene therapy, one that turns off a gene. As the name implies, gene inhibition or gene silencing is a technique that aims to reduce or eliminate the production of a protein from its corresponding gene. As we learned before, genes are sections of DNA that contain the instructions for making proteins. During normal protein production, the genetic code in the gene is copied into a strand of mRNA through a process called transcription. And then the mRNA is used to build a protein through a process called translation. So instead of directly editing DNA, the key idea behind gene silencing is intervening in the gene expression before translation. Essentially, by designing a molecule that can specifically identify and then break down the mRNA that is carrying the instructions for making a particular protein, scientists have been able to effectively decrease the levels of that protein. Gene silencing techniques have been widely used by researchers to study genes associated with a variety of disorders. These disorders include cancers, infectious diseases, respiratory diseases, and neurodegenerative disorders like, for example, Huntington's disease. Huntington's disease is characterized by a mutation causing excess CAG repeats in the Huntington's gene. These extra repeats are what lead to the production of the mutant Huntington protein, which results in the disease. So silencing of the mutant version of the Huntington gene is a potential therapeutic strategy for treating Huntington's disease. So gene therapy is arguably one of the most exciting areas of biotechnology at this moment. There has been a lot of progress recently in all areas of gene therapy, especially those related to gene editing, that are likely to unlock new areas of medicine over the next 10 years. So I hope you found the information in this lesson both interesting and thought-provoking. Talk to you soon.